The goal of my research is to understand the growth mechanism of protein aggregation. Protein aggregation is a biological process which protein molecules accumulate together to form a cluster. It is very often correlated with diseases. For example, Alzheimer's and Parkinson's disease are closely related to the aggregation of misfolded protein molecules. Protein aggregation is also a huge challenge to the pharmaceutical industry. Aggregation of protein-based drugs not only reduces the therapeutic efficacy, but more importantly, it causes the immune system response, which could lead to serious consequences of the patients. For all these reasons, it is very important to know how to suppress protein aggregates. The very first step to reduce protein aggregates is to understand how the protein molecules start to aggregate in the first place. In my lab, we use a new microscopic imaging technique to characterize protein aggregates. We shine a beam of laser on bovine serum albumin samples, and the aggregate in the sample solution scatters the laser beam and creates a hologram. The hologram contains the information of both how strong the light is and which direction the light is traveling to. The latter is a missing piece of regular microscopy technique. The analysis of a hologram tells us the size and refractive index of the aggregate. The refractive index is just a unique optical property for the material. From the size and the refractive index information of thousands of bovine serum albumin aggregates, we revealed that this type of aggregates has a fractal nature. So instead of filling up the three-dimensional space, the aggregates grows with branches, just like a tree. It only occupies a space dimension of 1.7, and a structure of 1.7 fractal dimension corresponds to a specific process called reaction-limited cluster aggregation. And the mechanism of this process is very well known to the science community. So the same methodology is transferable to understand the mechanism of other types of protein aggregates.